Bismillahirrahmanirrahim. Now we're going to do some difficult MCQs just before the exam, and I hope this is going to help you all in the exam taking place a few days from now. Question number one: microscopes vary in magnification and resolution. Which of the rows A to D below is correct? Now, if you have to look at light microscope and transmission electron microscope and scanning electron microscope, you have to realize this 1500 is the best magnification for a light microscope. So the only possible answers were these. But then when you look at the resolution is only 200 nanometers. It is not light microscope cannot magnify and show you all the details. Please do not say clear image. It's not a clear image. So the answer is C. But in order for you to revise this, I want you to look at these two diagrams. I want you to look at the important thing. Magnification 1500 is the useful magnification. But the electron microscopes are up to this. Now resolution 200 nanometer and 0.5 nanometer. So what you have to understand is you have to be really knowing that the difference between the light and the electron microscope. Another diagram for you to see. So this is another one which I want you to look at and see this. So please have a look at this and revise this part of the syllabus as carefully as possible. Now coming to question number 2, carbohydrates such as starch are made from monosaccharide joined together, glucose joined together. Which of the bonds A to D joins monosaccharides together? Now, as we all know, it is glycosidic bond. But then what would have been the answer if the answer was A, ester? The ester bonds are formed between glycerol and fatty acids, 3 fatty acids. So triglycerides would have ester bonds. Now, where would peptide bonds be formed? Peptide bonds would be formed in enzymes. Peptide bonds would be formed in hemoglobin. Peptide bonds would be formed in antibodies. Now you don't relate it, you say protein, but you don't remember the different type of protein, and that is what is going to cost you an MCQ. You realize you have to get 35 out of 40 in the MCQ to even score a decent grade. You have not taken paper 2 and 5. You are at a major disadvantage. So everybody must be very careful. Now phosphodiester would be where? It would be between in a DNA or an RNA. Or you could have said even a nucleic acid. So phosphodiester bonds would be in between nucleotides, and DNA has nucleotides, RNA has nucleotides, tRNA has nucleotides, mRNA has nucleotides. So rRNA has nucleotides, ribosomal RNA. So question number 3, which of the following statements A to D about the nature of the genetic code is incorrect? Now we didn't read this, is incorrect. If you do this in the exam, well, this is not going to be the correct way that we're going to handle this paper. Now we know that it is a degenerate code, it's a triplet code, it is universal. But what it is not, what is incorrect, it is overlapping. Overlapping means that if AAT codes for one amino acid, then CCG codes for another amino acid, but there is no overlap. This cannot be happened. And I want you to go through this paragraph which I have underlined, and I want you to understand this, because if there is a question on that in the exam, you must be able to do that. Now, I would like you to pause the video here and have a look at what I want you to read. I'm not going to read all of it, but I'm going to just read here. There are 64 possible permutations or combinations of 3-letter nucleotide sequences that can be made from the 4 nucleotides. Of these 64 codons, 61 represent amino acids, and 3 are stop signals. Although each codon is specific for only one amino acid or one stop signal, the genetic code is described as degenerate or redundant because a single amino acid may be coded for by more than one codon. It is also important to note that the genetic codes does not overlap, meaning that each nucleotide is part of only one codon. A single nucleotide cannot be part of 2 adjacent codons. Furthermore, the genetic code is nearly universal, with only rare variations reported. For instance, mitochondria have an alternative genetic code with slight variations. So I want you to read the whole of this paragraph, and I want you to pause the video here, have a look at it and understand this. Now coming to question number 4, which of the following structures are found in prokaryotes, which are bacteria, and in eukaryotes? Eukaryotes are animal cells and plant cells. So animal cells and plant cells are eukaryotes, and fungal cells. Now, wall made of peptidoglycan is only found in prokaryotes. Circular genomic DNA is found in bacteria. A nucleus surrounded by a nuclear membrane is not found in bacteria. So the only answer left is ribosomes. But why should B not be the answer? The answer is of course D. But why should B not be? In fact, the answer should have been B and D. Why? Because if you look at it, eukaryotes have mitochondria in them, and mitochondria have circular genomic DNA. So technically, this question is wrong. But I hope somebody picked that up. That technically, this question is wrong because circular genomic DNA is also found in eukaryotes. Because in eukaryotes, they have mitochondria. If it's an animal cell, it has mitochondria. If it's a plant cell, it has chloroplast, and chloroplast also has circular DNA. So just a discussion on the validity of this MCQ. This is a faulty MCQ. Now, question number 5. The pressure changes in one mammalian cardiac cycle are shown in the graph below. Now, this is the ventricular pressure. Let's draw it in red. So, this is the ventricular pressure is rising, and then it's coming down, and then it's going like this. And the atrial pressure, let's draw it in green. It's this one, which is very less minimal. So this will be atrial pressure. Which of the following time periods A to D show ventricular systole? You know, ventricular systole is when the ventricle is contracting. So that is this part of it. I would underline it by this part. It is this part. This is the time when the ventricle is contracting and the pressure is increasing, and here the semilunar valve will open, and at this point, at this point, the semilunar valve will open and blood will go into the aorta or the pulmonary artery. So the answer is, of course, between this time period, which is zero point 0.2 to zero point 0.3 seconds. So the answer is B. But you must look at this cardiac cycle graph and please revise it before you go for the exam. That's a very important graph. Okay, then we come on to question number 6. Pressure varies in different parts of the mammalian circulatory system. Pressure, blood in the aorta, tissue fluid, lymph, blood in the vena cava. Which of the following A to D correctly completes the table below? Now, which of the following options A to D correctly completes the table below? Now, the answer, of course, is C. Why? Because once the aorta has got the highest pressure, and then everything has to be less than that. So all of them would be low. It's a little vague, this question, but basically all the pressures are going to be low because so there was no medium, there was just high and low. So high would only be in the aorta, and the rest of it all would be, of course, lower than the aorta. That is why it's a sort of a little, I would say, a little changad MCQ. So the question 6, the answer was C. The image shows the stage in mitosis. Which of the following options A to D is the stage of mitosis shown above? Very easy question. You see, this is a cell. You can see this is the boundary of the cell, and this is right in the middle M metaphase. Now, please revise mitosis micrographs. You must revise this. You must have a look at the micrograph. Just Google it and search for it. Mitosis micrograph and label stage is labeled. So have a look at that so that you don't make a mistake in that. What does a prophase look like? What does anaphase look like? And what does telophase look like? Please do that. Then the diagram shows the simplified structure of an antibody. Which of the letters A to D identifies the region of the antibody that allows the distance between the antibody binding sites to vary? Now, some of you have problem understanding the English, and your English needs to be improved, and that is going to be your problem. So this is the distance which is going to vary. When is the distance going to vary? When is it going to become less? When is it going to become more? What is the important thing is the flexible region, is the hinge region. So the hinge region is going to change. So then this will spread out more, or it will become closer. So it says identify the region of the antibody that allows the distance between the antibody binding sites. These are the antibody binding sites to vary because this is the place where the antigen is going to bind. So antibody binding sites to vary. How is this distance going to be changed? How is the distance going to become more or less? It's the hinge area, the hinge region. So that is why question number 8, the answer is C. A group of students were investigating the diffusion of molecules across membranes using a model cell as shown below. Model cell, extracellular fluid, cytoplasm, solution containing starch and amylase. So inside the model cell we have starch and amylase. Biochemical tests were used to identify the type of molecules present. The results are shown in the table below. A tick represents a positive result. Which of the rows A to D show the correct results for the cytoplasm at the beginning of the experiment and the extracellular fluid at the end of the experiment? So you have to understand is starch is going to be digested by amylase. So what is going to come out is maltose. Starch cannot come out and amylase cannot come out. Now amylase is a protein, it's going to give you a positive test with biuret reagent. Amylase is an enzyme. An enzyme is a protein, it's going to give you a positive test with biuret. Starch cannot come out. Starch is going to give you a positive test with iodine. Maltose will give you a positive test with Benedict's test, reducing sugar test. Now you have to understand is that why would Benedict's test be present? Benedict's starch will be positive, amylase and all that will convert into maltose and the maltose will diffuse out. So the extracellular fluid will be positive. The biuret test will be positive inside the cytoplasm. Why? Because there is the amylase inside the cytoplasm. And the amylase is a protein, so that would be positive. And the iodine test would be positive because starch would be inside. But starch cannot come outside, so this will be negative. Similarly, the amylase cannot come out, so this will be negative. Similarly, the maltose, as it is formed, will be diffusing out into the extracellular fluid. So the answer is A. Which of the cells below represented by cubes A to D has a surface area to volume ratio of 3:1? So the answer is B, 3:1. Now I've done all the calculations. Maybe one or two of the calculations might not be correct, but the little that I could do, I have figured that out. I'm not such a maths whiz guy, so please forgive me if there's a mistake in it. A diagram of stained blood smear observed under light microscope is shown below. Which of the structures A to D in the diagram is a neutrophil? Now the answer is B. What is C? C looks like a lymphocyte, A looks like a monocyte, and D is a red blood cell. Then coming to question number 12, air moves in and out of human beings through the trachea, which is lined with cells. The diagram below shows a section containing these cells, cell X, Y, Z. Which of the following statement about tracheal cells is correct? Now, of course, we all know these. What is cell X? Is the goblet cell that produces mucus. Cell Y is the ciliated epithelium cell. And cell Z is the cells just below the ciliated epithelium cells. So the answer is C. Cell X releases mucus into the trachea. So this was the goblet cell which released mucus into the trachea. The rest of them are all wrong, and you can easily say why they are wrong. X and Y don't move the mucus. X doesn't move the mucus. Only Y moves the mucus. Cell Z is a goblet cell. That's wrong. That's not a goblet cell. That's one of the cells just underlying the ciliated epithelial cells. And these are all not columnar epithelium. These are actually stratified squamous epithelium. So that's a different name, but we don't study that in A levels. DNA is formed from three main groups of molecules: pentose sugars, phosphate groups, and nitrogenous bases. The bases can be divided into purines and pyrimidines as part of the syllabus. Identify the two purines. I usually remember it just like this: 14th of August, Pakistan Independence Day. So AUG, and so the answer is A. Either you know this or you don't know this. You're not going to really guess this out. You know that purines and pyrimidines, you have to know this. Purines are adenine and guanine, and the rest are pyrimidines, which is what? Cytosine, uracil, and thymine. Question 14. Which of the options A to D is the reason why plants require specialized transport tissue to allow osmosis to take place? Because they all have a large surface area to volume ratio? No, in fact, there's a smaller surface area to volume ratio. To carry sucrose to their leaves? The answer is D, because they overcome the limitations of diffusion over long distances. That is one of the main things that we understand, but this problem is not in your syllabus for 9700, but it's in other syllabuses. Many plants are adapted to the availability of water in their environment. One group of these plants is the xerophytes. Availability of water in their environment. Which one of the following statements correctly describes a xerophyte? You see, in a xerophyte, the main thing would be something to prevent water loss. And the answer is C. The poison tree has leafless branches covered in thorns to reduce water loss. But basically, what you have to understand is why are these green? These are high saline marine estuary, but that's nothing to do with xerophytes. They are the ones which survive in desert conditions. The water lily has air and camera to allow movement to submerge root. That is hydrophytes actually. And these are, of course, nothing which are complete its entire life cycle submerged in shallow ponds. You see, xerophytes are those which survive in deserts. So all these were wrong and C was correct. Then which of the options A to D is correct statement about polysaccharides of glucose? A. Cellulose microfibers are formed by hydrogen bonding between adjacent chains of alpha glucose molecules bonded with 1,4-glycosidic bond. Now you know cellulose does not have alpha, it has beta. I told you to remember it like BC before Christ. So beta is cellulose. Amylose is a straight chain of alkyl glucose monomers bounded by 1,6-glycosidic bonds to allow for dense packing. Now that is wrong because amylose has 1,4-glycosidic bonds. Glycogen has a high proportion of 1,6-glycose to produce a highly branched molecule. Yes, that is the answer. Amylopectin has a mixture of 1,4 and 1,6 between beta. Now here, this was the mistake. This is wrong. Beta glucose, alpha glucose. Amylopectin, amylose is amylose, amylopectin, and glycogen is alpha glucose. Only cellulose. Only cellulose is beta glucose. Please remember that. Only cellulose is beta glucose. Question 17. A group of students was given a 1% solution of an unknown digestive enzyme. They were also given 3 tubes containing an identical mixture of foods. The students carried out different biochemical tests on each tube before and after adding the unknown enzyme. The results are shown in the table below. Purple remained purple, so that means the protein was still there. Nothing digested the protein. The reducing sugar was still there. But the iodine test, the blue-black, changed to yellow-orange. Now, name the type of enzyme the student used. Carbohydrates. It was digested the starch. Starch is a carbohydrate. Question 18. DNA carries the genetic code which is non-overlapping and degenerate. Which of the options A to D contains the correct definitions of non-overlapping and degenerate code? Each nucleotide is only part of one triplet of bases and the molecules break down easily. The genes follow straight after each other and the molecule breaks down easily. Each nucleotide is only part of one triplet of bases and more than one triplet codes for a specific amino acid. The genes follow straight after each other and more than one triplet codes for a specific amino acids. Now what said? Which is the correct definition for non-overlapping and degenerate? The answer is C. Each nucleotide is only part of one triplet of bases and more than one triplet codes for a specific amino acid. So non-overlapping and degenerate. And question number 19. An individual bitten by a rabid dog can be treated by an injection of human rabies antibodies. Antibodies, passive immunity. Which of the option A to D describes the type of immunity provided by this treatment? You see, when you give antibodies, it can be either be natural passive or artificial passive. And because you are giving them antibodies, it can be treated by an injection of human rabies antibodies. So we've made the antibodies in the lab. So that is why the answer would be artificial passive. Natural passive would have been if it was from mother to fetus, either across the placenta or in breast milk. Now coming to question number 20, a student studied the structure of a blood vessel and found an outer layer of collagen fibers, a thick middle of smooth muscle and elastic tissue, and innermost layer of endothelial cells. Which of the options A to D identify the type of blood vessel the student studied? Of course, the answer is A because smooth muscle is only found in arteries. Smooth muscle, a thick middle layer. So that will only be an artery, that can't be a vein or a capillary. It of course has nothing in it. It just has one cell thick endothelium. Endothelium, please remember that. Which of the option, question 21, which of the option A to D describes the role of cholesterol in cell surface membranes in the human body? Cholesterol binds to phospholipid phosphate heads, increasing the packing of the membrane, therefore reducing the fluidity of the membrane. Cholesterol binds to phospholipid fatty acid tails, reducing the packing of the membrane, therefore increasing the fluidity. Cholesterol absorbs ATP, preventing active transport across the membrane. Cholesterol binds to phospholipid fatty acid tails, increasing the packing and reducing the fluidity. So here it was increasing the cholesterol binds to the phospholipid phosphate heads. So just read this very carefully and then think about it and then decide what is going to be your choice of the answer. You see, what you have to understand is why was the answer D? Cholesterol binds to the tails, increasing the packing of the membrane, therefore reducing the fluidity. When it becomes very closely packed, then it's going to reduce the fluidity. Fluidity means the ability to laterally move. But if it's going to be very packed together, then how is it going to move? Just like if three people sit at the back of the car or four people sit, well, how are they going to move then? So that is why the answer was D. Question 22. During DNA replication, DNA polymerase can only work in one direction from the 3' to the 5'. This means that the lagging strand has small gaps left in the backbone. DNA ligase works to seal these gaps. Which of the option A to D identifies the bond formed? You see, the gaps is between the DNA. So there is this gap in the lagging strand. And those little pieces are called the Okazaki fragments. So what is going to join them? What is going to join them is the phosphodiester bond. Nucleotides are only joined by phosphodiester bond, not by hydrogen or glycosidic bond. It's the gaps left in the backbone, in the sugar phosphate backbone. But hydrogen would have been answered by most of the Changar students because they would have said, hydrogen bonds, miss A and T के बीच में hydrogen bond होता है. And that means you've not understood. You're not even understanding the question. But of course, we'll always have a few Changars in every class. Which organelles A to D is not involved in the production and secretion of enzymes in eukaryotes? You see, enzymes you've got to remember have to be made on the ribosomes, and then they form the secretory vesicle, and then they go to the Golgi, they're modified, or a glycogen part is added, and like pepsin, pepsinogen, and then all that, and then it's going to be thrown out of the cell if it's an extracellular. If it's an intracellular, well, it's not going to be thrown out. So you have to understand is that the smooth endoplasmic reticulum does not take part in any. That's for lipid synthesis. 4 autoimmune diseases are often treated with the course of antibody injections. Which of the following A to D describes immunity arising from this treatment? So whenever you give injections would be D, passive artificial immunity. Passive, if you give antibodies, natural would be mother to fetus. If it's injection, then it will be passive artificial. The image below shows isomaltulose, a disaccharide formed from alpha glucose and fructose. Name the bonds that hold the alpha glucose and the fructose together. 1,6-glycosidic phosphodiester bond, ester bond, 1,4-glycosidic bond. Now you can see here why this would be a 1,6-glycosidic bond. So you see, if you number them 1, 2, 3, 4, 5, 6, and this is then 1, 2, 3, 4, 5, and then 6. So you have to see it's a 1,6-glycosidic bond. It was an alpha glucose and a fructose. Phosphodiester is between nucleotide. Ester bond is in fats. 1,4-glycosidic would have been a different bond. So please revise 1,4 and 1,6-glycosidic bonds. The video 1 will have these questions. We'll continue the rest. There are another 20 questions to be done. We'll continue this in the second video. Thank you very much.